the lake study. It just started this past summer. It's going to be going on for the next couple of years. So they're here to kind of give us some information, an intro about kind of the scope of the study, some of the methods that they're going to be using over the course of the next couple of years. I think that's pretty much going to be the extent of it. One thing I just want to remind everybody, since it is live streamed and we're recording this for folks that can't come, is to please just save your questions till the end, and then we'll have time for everybody to kind of come up to the mic and ask their questions. But with that, I'll let these guys go ahead and get started. Thank you.
All right, well, thanks, Ray, for that introduction. Can everybody hear me okay? Yeah. Great. So as Ray mentioned, I'm Dr. Troy Farmer from Clemson University, work in the Department of Forestry and Environmental Conservation. And I'm a trained fish ecologist and manager. My program at Clemson University really focuses on all things aquatic, fish included, and really excited to be here. We appreciate the invite to come up and work with you all at Conestee. We've started working on the project during late summer, so today I'm going to be giving you, get some feedback. Gotcha. A little better now? Yep. Excellent. I will stay back here. I tend to walk around when I talk sometimes, but I'll keep that behind this area. So really have enjoyed being up here so far. We've started collecting some preliminary data already for the lake study, which just started this fall. So today I just have a brief presentation to give everyone here kind of an overview of the goals of the project, the types of data we'll be collecting, and hopefully the type of information we'll be generating for you all that'll be informative for your management of these systems. So I'll give that in the first half of the talk, and then I'll turn it over to Dylan Windricks, who's the master's student working on the project. If you guys live on the lakes or spend time around the lakes, you've probably seen Dylan up here. He's been leading crews of our undergrad students from Clemson in some data collection already, and Dylan will continue to be up here over the next year and a half or so for the full 2 years of data collection to continue leading that effort in the field. Dylan's going to give the second half of the talk today, which will cover some of the preliminary data he's collected on the fishery side of the study to show you all some of our initial information for fisheries and what that looks like. Let's see here if I can get this to advance. Just have to click on the presentation. Gotcha. Arrow keys work excellent. So today's outline, I'll just go over some background information, get into the objectives of the work, what we hope to accomplish, and talk about a timeline of activities. Then I'll turn it over to Dylan for preliminary results, and then we hope to have plenty of time for questions and discussion. So just a brief intro, and then lots of time, hopefully, to get questions from you all, and also would value some feedback from you all. Just every time we come up and chat with people, we learn more about the history of these lakes and the systems. So hope we have plenty of time for those discussions today as well, as we're certainly learning from you all as well. And this is a two-way street today. So just a brief background. Of course, lakes provide really important, what we call as scientists, ecosystem services. And those can include things like provisioning, such as fisheries resources that might have a distinct economic value to them. Ecosystem services can also include things like cultural and recreational value that improve the quality of life, right? And so that can include things like swimming, boating, fishing, and also just the general aesthetics. There's lots of scientific studies that show that for people that spend time around natural ecosystems and in the outdoor environment, that general health outcomes are improved. And you can actually, a lot of studies have put dollar values to this. So these ecosystem services that the lakes here at Conestee are providing, certainly I think most in the room would agree, provide a suite of ecosystem services that are extremely valuable to the community as a whole. For those ecosystem services to be maintained, of course, sound management is required. Whenever we have human development and activities on the landscape, water runs off our landscape into our lakes, and so sound management of our watershed is required to maintain healthy lake ecosystems and maintain those ecosystem services, the fisheries, and the other benefits that those lakes provide. From conversations, I understand there's been a long history of management here at Conestee, and I think that kind of led up to, in the fall of 2024, the Conestee Falls Lake Management Subcommittee developing an RFP to study the 4 reservoir ecosystems. And that RFP really requested a proposal that would address 4 key points. Those were watershed characteristics, reservoir habitat, water quality, and then recreational fisheries, all with the goal of collecting data in these 4 areas, and then that data would inform management of the ecosystem as a whole. Understanding that the watersheds around the lakes are just important to manage as the lakes themselves. So in response to that, my lab at Clemson University submitted a proposal that was evaluated and eventually selected for the project, I guess in early 2025. We then initiated the project this summer of 2025. Dylan arrived in Clemson this August, and our sampling really began kind of late summer and fall. So we're just now getting started on the project and have at least a little bit of preliminary data to share with you all today. The project, as outlined in the proposal, really contains 3 distinct objectives. The first is to conduct a comprehensive assessment of reservoir water quality. That's things like temperature, dissolved oxygen, conductivity, pH, kind of our physical parameters of water quality. Also a survey of submerged aquatic habitats. That includes things like coarse woody debris, physical structures that are in the lake that fish might use, and important features in the surrounding watersheds that also affect reservoir hydrology and quality. So the first objective is kind of understanding reservoir water quality and watershed characteristics that affect reservoir water quality. The second objective really focuses on the fishery resources of the lake and to conduct standardized fishery surveys to assess what fish species are in the lakes, the food web structure, and how those lakes can be managed to enhance the fishery, to increase value of the fishery to the community. And finally, our third objective is to produce a final lake management plan that summarizes findings from both water quality, habitat, and fisheries surveys. And included in that lake management plan will be some specific strategies for watershed, reservoir, and fisheries management. So the end goal here is really conduct science to inform sound management of the ecosystems here on Conisty Falls. The aquatic ecosystems, I should say. So just to tell you briefly a little more about each aspects of these objectives, I wanted to talk a little bit more about each of the surveys we'll be conducting. The first one is a habitat survey. That'll include a watershed survey where myself and students will actually physically survey most of the property that drains into the reservoirs, most of the property immediately around the reservoirs to identify all incoming streams and inflows into the reservoirs. We're also going to utilize GIS mapping and publicly available maps of land use, land cover for the community to understand what different watersheds for each of the lakes look like and how that might contribute to sources of runoff, sediments, and nutrients coming into each of the lake. The goal of this is to really identify important areas for sediments and nutrients that are coming into the lake. So if those are deemed to be excessive, those are the areas where remediation efforts would need to be targeted. And again here, the goal is to not implement, but to provide recommendations for management practices that can improve water infiltration. Of course, when we have water infiltration into the ground, that absorbs sediments and nutrients and keeps those sediments and nutrients from running off into the lake. And generally, as most of you know, these lakes are clear, cold water, low conductivity. For their natural setting to be maintained, they need lower levels of sedimentation and nutrients. So the goal of the watershed survey is to inform that sediment and nutrient management across the landscape. We'll also be conducting an in-lake, what I'm calling a reservoir habitat survey. And just to note, I'm using the term reservoir for our lakes. I'm just using that because they're man-made systems created with the dam, but lake and reservoir you can use interchangeably here to talk about these systems. We'll have an in-reservoir habitat survey. We'll be conducting an aquatic plant survey. Dylan and some students and myself will be getting out and actually conducting some snorkel surveys along transects to identify species and baseline abundances of native and invasive plants that might be in the lake. And then we'll also be conducting a physical habitat survey to look at things like coarse woody debris, that's large trees and wood that's in the system. That type of physical structure is very important for fisheries. And making some recommendations there. We actually have some set metrics from the literature that we know productive bass and bluegill fisheries require a certain density of coarse woody debris to enhance their productivity. So we're interested there to see where the lake stands, if it needs more structure, or if there's sufficient structure for cover for the fisheries the lake supports. We'll also be quantifying rock substrates and key areas of sedimentation in the lake, all with the goal of informing management of habitat in these lake ecosystems. Dylan and his crews are also conducting seasonal water quality surveys. That's using our YSI meter to measure nearshore and offshore depth profiles. So Dylan's going out in a boat, taking what's called our YSI instrument, which is a digital meter that measures temperature, dissolved oxygen, conductivity, and pH. And he's taking those readings at each meter, taking depth profiles at multiple spots in each lake so that we can get an idea of seasonal water quality dynamics in the lake. This study especially has a real emphasis on thermal stratification. We know all lakes in this part of the world here in the Southeast thermally stratify during the spring and summer, which means the warm water on top actually does not mix with the cold water on bottom. It's just like oil and vinegar, where you have that warmer water, which is a much lighter density, and it sits on top of the denser, colder bottom waters. That type of thermal stratification is normal, but when it's prolonged, eventually we can get oxygen depletion in those bottom waters that don't remix with the atmosphere. So Dylan's carefully measuring this on a seasonal basis to try to understand the thermal dynamics of the system. As you all can imagine, that's very important for fish habitat, growth, production. And so those physical temperature structure of the lakes is really important for understanding their potential for fish production and understanding fish habitat within the lake. Within the lakes, we have species like largemouth bass and bluegill that like that warm surface water. There's species like rainbow trout that like the cold bottom water. So understanding habitat availability for species-specific recommendations is really important moving forward. We'll also be taking some zooplankton surveys. So these are surveys of the tiny microscopic animals in the lake. You really need a microscope to see these creatures, but they're really important at the base of the food web. Zooplankton form a key food resource for larval fish. That's very small fish right after they hatch out. And without sufficient zooplankton densities, you can get very poor survival of larval and juvenile fish, which can, of course, limit your production of larger fish in the future. So Dylan and his crews will also be taking some zooplankton surveys starting this spring, looking at densities of what we call calanoid and copepod zooplankton, which are common groups of plankton that are food for fish, to understand where these lakes are in terms of their densities for larval fish food, which can help us understand their ultimate fishery productivity, or I should say their potential for fisheries productivity. So that's our habitat surveys. And that brings me to kind of the last type of survey we're going to be conducting, which are our fisheries surveys. You all may have seen Dylan out doing some fishery surveys. We've already started this work in the late summer and fall. And the main goals of these surveys are to collect fish to understand, number one, what species are in each lake. Number two, what the size structure of bass and bluegill and sunfish populations is. Size structure is a really important metric. If you have all young fish, that might not be so great because you don't have enough large fish for anglers to catch. It might indicate that something might be limiting growth in the population, right? If you have a population of stunted small fish that's not growing to larger sizes. If we have a population that's all larger fish, that may seem great, and it probably would be for a while, but without any smaller, younger fish coming to grow into larger sizes, you might have recruitment failure and lack of reproduction, which means the lakes are great now, but you might have trouble coming down the road, right? So ideally what we'd like to see for size structure is a range of sizes. Where you have good growth, fish are reaching larger sizes. We have good recruitment, which is what we call the reproduction of the next generation. We have smaller fish coming into the population, and we see the population that's self-sustaining with good growth and reproduction and equal representation across many age and size classes. So our analysis of size structure is really important. Ideally, in a system that's largemouth bass and sunfish dominated, like the lakes here are, we really go back in terms of managing that system to some very basic size structure indices that Dylan is going to be presenting today, which help us tell if the lake is in balance. And when I say balance, I mean if it's balanced between its predator and prey populations. So for a lake to have really good fishery productivity, you need a fair amount of prey, and you also need a high amount of predators that can control that prey population. If our prey population gets really high and our predator population is low, those prey can actually consume the juveniles of the predator and suppress the predators. And that's called prey or bluegill crowding, where you have a lot of bluegill and actually not many largemouth bass because the bass are being suppressed by the bluegill. You can also have predator crowding, where you have too many predators and not enough prey. And that one's easy to understand because when you have a lot of predators, they'll suppress the prey population and then essentially limit their own food resources. Growth will be low and survival will decline. And so when we say a pond that's in balance, we mean it has the right amount of prey for the predators and that each are in equal density that support good growth of the other and good reproduction and a healthy, productive fishery. And that all gets back to size structure indices that were developed years ago by Dr. Homer Swingle at Auburn that are kind of fundamental to our understanding. So Dylan's going to be presenting some data today and talking about size structure and some initial reads on where the ponds are in terms of their balance. So when you hear Dylan talk about balance, that's really what he's talking about, is predator and prey balance in these systems. We're also going to measure condition, which is just getting a fish's length and weight to look at how skinny versus how plump a fish is. That tells us if the fish is eating well. Just like us, when you go to the doctor and get that BMI index, mine's always a little higher than I want it, right? That body mass index. But we're kind of going to do the same thing for fish. That basically tells us how plump or how skinny they are, and that's also a good read on is there enough food in the system. If we have a lot of fish swimming around there too skinny, that might indicate a lack of prey in the system. We'll be collecting data, again, abundance is just how many of each species are present, and then community composition. We're going to use a diversity of gear types to collect all of this fishery data. We're going to use standardized daytime and nighttime boat electrofishing. Some of you may have seen us out on the lakes. We've already started this collection of the boat electrofishing data. This is a really common standardized way to collect fisheries data. The system that we're using is built by Midwest Lakes Electrofishing Systems. It has a high control of the frequency and the pulses that we're putting into the lake, and we're following published guidelines to target the specific species that are present in these lakes. Electrofishing is non-lethal for fish, so it stuns them and temporarily puts them into a state of what we call electronarcosis. It kind of stuns them. They float to the surface. We can easily net them, put them in our live well, get a length and weight on them, and then by about the time we're releasing them, they're ready to go. They're waking back up, and they're going to swim away unharmed. So it is a non-lethal standardized technique. The folks at North Carolina Wildlife Resources Commission that manage all the public resources in North Carolina use the same exact standardized equipment and protocols that we're using for these surveys as well. We're also going to be using hoop nets, primarily that target catfish. Boat electrofishing doesn't do a great job of collecting catfish, so we're employing some hoop nets to get catfish data. Fyke nets, which are also a passive net, those have done well for collecting rainbow trout, which are a little deeper than our electrofishing tends to sample. And then seining, which is when Dylan and his crew put on waders and then drag a seine net, which is about 12 feet long by about 4 feet high through the shoreline areas. And those seines are really effective at collecting our baby fish, the juvenile bluegill, red ear sunfish, and largemouth bass. And that's a really important read of reproduction. So we want to see a good abundance of juveniles across size ranges for all the species we care about. So if you've seen Dylan trekking around, if you got a lakefront home and he shows up with waders and a seine in front of your home, he's just there getting a read on fish reproduction and trying to collect some data there. So I'll also note these lakes for the Southeast are really unique, and you all probably know that. A lot of what we know from fisheries management is collected at lower elevations with lakes that are warmer and more productive than these lakes at Conisty. So a lot of the fisheries management recommendations that we have were developed for systems that are a little bit different from yours. And I think that difference is probably what led to the study that we're currently conducted being needed, because I think for years, kind of standardized fisheries approaches have been tried to be implemented in these systems with maybe a mixed bag of success, as I understand it. So hopefully the next part, the diet study, will really highlight where these lakes are unique. To really understand a system and manage it, you need to know how the food web is connected, right? And so we'll also, as Dylan collects species, be taking diets from those, primarily from our predator species like largemouth bass and rainbow trout. to understand what the predators are actually eating in the lake. Is it sunfish, or is it maybe macroinvertebrates and salamanders that are living in the aquatic vegetation? Anecdotally, we've already seen some of that, so we know it's happening from the limited diet data we've collected already. But the diet data will really help us understand exactly how these lakes are put together from an ecosystem perspective and what the important prey resources are for the fisheries that we'd like to manage. The final component of the fisheries survey is a largemouth bass mark recapture study. And that's where Dylan is going to be putting in some small individual tags in each bass. This uses the same RFID technology where if you're in Walmart and walk out the door with an item and they forget to deactivate it, it beeps at you. Dylan's putting these small pit tags in largemouth bass. When we scan that fish with our wand, it'll give us a unique 16-digit ID for that specific fish. So we can track fish over time and tell if we've seen that fish before. If we've seen it again, how much is it growing? Is it surviving? And also that data will help us estimate population size using some basic kind of ecological statistics. So we're also going to get a population size estimate on the largemouth bass population for each lake, and then use something called bioenergetics modeling that really just estimates energetically what the largemouth bass population would need in terms of food. So what bluegill densities would need to be in the pond to understand the consumptive demand of the largemouth bass population size. The goal of this modeling, and why we're going through all this trouble, is to really understand if the lakes have adequate prey resources to support a healthy largemouth bass population. And if not, what supplemental stocking of prey resources might be needed, right? How many should we be thinking about putting in so that there is enough prey to meet the consumptive demand of the largemouth bass population in the lake? So that's that final component that's really focused on the largemouth bass, which from our understanding support an important fishery in the lake as our top predator, as is the case most of the time in our southeastern reservoirs. So just a general timeline of some project activities. I know this is a little bit small, but on your left you have 2005. The type of activities we'll be conducting, and you can see the survey began this summer with some preliminary surveys where we came out, took water quality on the lake, conducted some preliminary fishery surveys. Dylan, then when he arrived in August, shortly thereafter in September and October, has conducted our first round of standardized fishery surveys this fall. The data he'll be showing are from those collections he conducted this fall. We've also taken some detailed water quality surveys starting this fall. And here we are today at our project review meeting for year one. So that's here this winter. And then you can see moving forward, we'll be conducting fishery surveys on a seasonal basis for the next 2 years. Physical habitat surveys next summer in a very intensive physical habitat data collection period. And then the goal is that the final lake management plan will be produced in summer of 2027. And that will be followed up with some meetings to talk about specific management recommendations that we would recommend based on the science we've conducted on the lakes to improve both the fishery and watershed management of the systems. Of course, I've got Dylan's final master's MS, that's the Master's of Science thesis on here as well. This project's going to form the basis of Dylan's master's work, so he'll be wrapping that up in December of 27 after he completes the final lake management report, and then of course has lots of feedback and discussion with this group to kind of make sure everyone's on board with those management recommendations. All right, I think that's it for my portion of the presentation. So for the rest of it, I'll turn it over to Dylan, and he's going to talk a little bit about some preliminary fishery data that he's collected. So
Sounds
Dylan?
great. Thanks, Troy. So hi, everyone. I see a lot of familiar faces out there, but I'm Dylan Wendricks. As Troy mentioned, I'm the master's student on this project. I'm in the Wildlife and Fisheries Biology program at Clemson. A little background about me. I recently completed my undergrad at University of Wisconsin-Madison. My degree was in environmental science. University of Wisconsin has a long history of, it's known as the birthplace of limnology. So I have a lot of background in working in lake systems, also in fisheries research as well. Troy poached me from the Wisconsin Department of Natural Resources, where I was a technician there, working with their fisheries research program. So today I just wanted to touch on some of our preliminary results that we've been able to collect so far. Sorry that graph there is a bit blurry, but I just wanted to show this plot here. This is a bit of a fish community snapshot Big takeaway you can kind of pull from this is that across reservoirs, it's a relatively similar fish community. You have your largemouth bass, your bluegill, your other sunfish species, rainbow trout and catfish are also present in all reservoirs. Atagahi stands out a bit where there's a population of yellow perch and crappie as well that are present. And then we also have mosquito fish that are found across reservoirs, and also we found some swamp darters, which are, and those last two there are non-game species, so they're smaller body minnow species. And to kind of jump into some of the other metrics that we've been deriving so far. As Troy mentioned briefly, we were looking at condition analyses for a few of the game species across reservoirs, and this graph here shows the relative weight of largemouth bass. So as Troy mentioned, it's kind of like a BMI indice. And 80 to 90 is kind of that cutoff of whether a fish is healthy or not, I would say. So this fish here out of Toccoa was particularly in poor condition. So that one was probably like in the low 70s range for relative weight. But relatively consistent across reservoirs, not particularly low either across reservoirs. And as you can see, relatively large standard deviation bars there. All preliminary data. We're hoping to kind of narrow some of these matrices in as we conduct our survey. This here, similar plot, but for bluegill. So there's a picture of a bluegill from Toccoa with a very high relative weight. So that was probably in the upper 90s. So you can kind of see the difference between the two species there. And then also for our bluegill condition across reservoirs, we're seeing a bit of a trend, or it's not a trend, a difference between reservoirs. So you can see there, Tioga, it's a bit lower. But again, this is all preliminary results. We're hoping to kind of refine some of this in more, but it does seem like the bluegill across reservoirs have a bit of a better relative weight compared to the largemouth bass populations. And then also, as Troy touched on a bit here, we've been calculating we've been looking into the size structure of largemouth bass. So you can see here, the x-axis is in total length of each individual fish, and the y-axis there is a count, or a number of individuals that fall within those length bins. So you can see for the majority of, I guess for all of the lakes, there's a bit of that bell curve shape where there's a high density of fish within that 8 to 12 inch range. And then they kind of taper off as you get larger and smaller, which is as you would expect across reservoirs and through systems. And also this red line here that I denoted in the graphs, you can see that's right at the 12 inch mark, which is, as Troy mentioned, kind of the Swingle indices, or it's kind of getting at this measure of balance that we're hoping to understand between predator and prey species. The 12 inch line will come into play in the next slide, but you can see in Atagahi there's a very large proportion of fish that are across that 12 inch line, whereas for the other reservoirs are all slightly below that, around 10 or 11 inches is the kind of the peak of that bell curve. So I guess with this plot, which I know can be a bit overwhelming at first, but stick with me. Kind of the key that you're looking at here is these lakes that are falling in this upper left quadrant is bass crowded, as Troy kind of alluded to earlier. And this bottom right quadrant would be bluegill crowded. And these axes here dictate, it's a metric called proportional size distribution. So you can think of it as a measure of the percent of the population that is above and below that 12 inch mark, essentially. So when you have a reservoir like Atagahi, for example, where it seems like this population has that mode, that peak that's across that 12 inch line, you end up getting a largemouth bass PSD down here on the x-axis. So that kind of brings it into this balanced quadrant of what we would assume. Whereas for Tioga and Wantesca, that largemouth bass population is significantly smaller. So it ends up in that bass crowded, or what we would expect a system to be bass crowded would have those characteristics. And then Toccoa is somewhere in between those systems there. But it's kind of a crude indice of these population dynamics. So we're hoping to kind of refine that further as well as we collect more data. And then Troy also touched on this a decent bit, but this will be kind of the next step into our project would be an exact abundance estimate. So picture there on the left is a pit tag transponder. So it's a small, not even half-inch long glass unit that we insert into the body cavity of the fish. So here you can see this mark on this largemouth bass. That's kind of the entry point. We insert it surgically with a needle, just like taking a shot. So we'll enter it into the body cavity of the fish, and then once you recapture a fish, we can read that unique code that it gives the individual. And I would digress not to include a picture of me with a big largemouth bass that we've sampled. But for example, that fish has a unique code in it. Next time I catch it, hopefully, I'll be able to know that it was that individual that I ran into in November of 2025. So with that, we wanted to open up for questions and discussion section, unless.
Yeah, turn it back on. So yeah, so feel free to come on up to the mic and ask questions.
Hey there, I'm Michael Solomon. I live on Lake Witeska. Had a home on Witeska for 28 years, so I've seen change over the years. I have two questions, if that's okay.
Absolutely.
All right. The first question I see in scope here is reducing sediment is something you're going to be looking at. As you've probably learned in your background for this research, we've had sediment removal from the lakes in the past, and the sediment comes back. So is within scope specific recommendations on how to reduce sediment and maintain those necessary levels of sediment? And how will you address the fact that a lot of the sediment comes from private properties?
It's a great question. I will say sedimentation is a natural process in all our reservoirs. Anywhere where water slows below a velocity, it certainly drops out the sediments it's carrying. So it's definitely a natural process. But you're also right that human activities on the landscape can certainly increase the amount of sediment that water carries in it. So we will be, in our survey of the watersheds, identifying what seem to be important inputs of sediments. We'll be gauging that from identifying key inflows into the lake and then assessing key areas of sedimentation in the lake visually from some snorkel surveys. And we will be making some management recommendations for things like if there are, aquatic or terrestrial plants that can be planted in the watershed, there's a lot of techniques. I actually teach a watershed management course at Clemson. We have a whole manual that Clemson Extension's developed on this. But there's a lot of techniques given your setting that you can apply that'll help slow water down, that'll help water infiltrate into the soil where those sediments will be picked up and intercepted. So we certainly anticipate providing specific management recommendations for each lake. And those will just be recommendations. There'll probably be a suite of recommendations with the understanding that cost is always limiting to what you can do. But those will be spatially specific for each lake.
Just to clarify on your answer there, when you say additional vegetation, might that be in the streams that are feeding the lakes? Is that what you're talking about?
Usually along the stream banks, that's correct.
The stream
to kind
banks.
of intercept water
Gotcha.
just as it gets to the stream before it flows into the stream.
Okay.
Yep.
My second question, you talked about recommendations on vegetation. All of our lakes have berms. And those berms, there's a couple of challenges there. One is sometimes if there's a tree or bushes on the berm, if they get too top heavy, they disrupt the berm, so they have to be taken down. We've had a lot of vegetation that's been removed that has not been replaced. Habitat ideally likes to have some vegetation overarching onto the lake water so that, like you talked about, the young ones have a place to hide and so forth. Is that within scope of your study, is to recommend what to do in terms of vegetation around the berms?
Sure, I hadn't gotten that specific, but we can certainly, if that's a concern, we can certainly look at that. You're right that complex physical structure is really important for juvenile fishes. And that's something we'll be kind of quantifying in these surveys, is how much complex structure there is for small fish and how much coarse structure there is for large fish. So if that's lacking, then we'll certainly be making some recommendations on how maybe that can be improved.
Okay, thank you.
Yeah, thank you.
Rich McCullers, look here, says 2021, Fish the Lakes. It might be early for this question. Have you done any initial surveys on the food chain, such as the phytoplankton and the zooplankton, or is that coming later on in the year? Because you gotta have the base of the food chain. Have you done any initial surveys on that?
That's a great question. Thanks, Mitch. We have not. That'll be starting this spring. That we'll be looking at some of the plankton dynamics of the lake. We won't necessarily be getting into the phytoplankton species, but we do have some techniques. We can look at chlorophyll A production, which just tells us how much phytoplankton. So quantity we'll be looking at, and then Dylan will be looking at plankton surveys, zooplankton, for the microscopic animals, the copepods, the rotifers that are filtering that phytoplankton and providing food to the fish. So they'll be coming this spring, but we don't have any data
That's fine. I
yet.
thought it might be a little bit early.
Yep.
Follow up to that one too there. Okay. But, so we know that the lakes are populated by fish that have been stocked. Where do the phytoplankton, zooplankton come from if they're not, are they just brought in with the fish, or how did they get there to begin with?
That's a great question. So, in systems like these that were originally streams, there are definitely some natural phytoplankton and zooplankton that were in those streams that probably colonized the lakes as soon as they were built. For most of that, it can, phytoplankton can get transported on droplets of water. It doesn't take much. So birds that are wading birds, like great blue herons, can move some of those droplets around. Some zooplankton species, I don't know, we haven't done plankton surveys yet, have resting age egg stages where they can actually have some level of desiccation and survive. It's really species specific. That allows those species to also get moved around by other aquatic organisms that, like birds or terrestrial or amphibians that might be moving. So there's a lot of potential ways that these things, even though they can't themselves move between systems, they can get moved in. But there was probably a natural community that when the reservoirs were built, selected for species that were a bit more tolerant of standing water instead of flowing water. That's a great question, yeah.
Ron Phillips, 12-year resident, very avid fisherman. You talked a lot about bluegill as far as the primary food source, the small bluegill. Is any part of this, your recommendation, going to be what other sources of bait fish that we should be putting into these lakes? Because I fish all these mountain lakes, and bluegill is not the primary forage fish. It's usually a threadfin shad, blueback herring, something like that. And I know there's been a lot of investment in the past of the different minnows that have been put in this lake, but really knowing what we should be putting in this lake to have the best survival rates and develop a sustainable population.
That's a great question. So some of the work that Dylan talked about with his pit tagging, I think I got too close to you there. Stay right here. Dylan's going to be developing some population estimates for largemouth bass, which will get at the consumptive demand of the population. I think the first question is there enough natural forage to support the largemouth bass through the sunfish populations of bluegill, red ear? If there's not, then I think the recommendation will be that you as a community might need to think about supplemental stocking of some sort of forage. That's difficult because sometimes those threadfin shad are great, but they typically don't naturally reproduce in these reservoir systems, so it requires an annual stocking, which can become costly. So certainly we will be thinking about those types of questions and what the right prey species may look like if the bluegill population seems like it's not productive
I
enough.
know the blueback herring spawn, and that's maybe deceptive. Blueback herring is not a herring, actually. It's actually a shad. I mean, I fish the herring spawn on these lakes. I know where it takes place, so I know that's a sustainable fish.
It is. Our blueback herring populations certainly naturally reproduce, and if there was a supplemental stocking, I don't want to get too far ahead of ourselves, but that might be a better species to think about.
Great,
Yep.
thank you.
Sure.
not tall enough. Mary Freudenberg, I just wanted to say thanks so much, Dr. Farmer and Dylan, for doing the presentation today. Just one follow-up question. In one of the slides that you presented where you had all the different species of fish in each of the lakes, I think there were 3 species that were unique to Atagahi. I was just wondering if there's something unique about Atagahi that would justify having those additional species that aren't in any of the other lakes.
Great question. Since you've spent so much time on the lakes, you want to put you on the spot?
That is a great question. It's something I've been thinking about. I honestly cannot tell you at the moment. I think looking at the stocking records, I think that the perch and the crappie population there is likely directly from that. But we're looking at and again, the seasonal water quality data might be able to tell us, something more there. And also the parrot feather introduction and that the greater abundance of macrophyte coverage might also be playing into this. But there's a lot of things that might be leading to that we're not totally sure yet. I'll say.
Thank you.
Hi, Jack Lang. I've been a homeowner here since 2006. Speak of the microphone, please. Sure. My question is about the spotted bass. Have you guys, are you grouping those in with largemouth, or are you separating those out? Because it would seem to be very difficult to do a study separating those out. Because I noticed you didn't include that in your species list.
You're exactly right, and that's a great point. We probably should have spent more time on it, so I'm glad you asked that question. We are identifying each bass at the species level, so if it's a spotted or largemouth.
Okay.
And the vast majority of fish we have seen in our boat electrofishing surveys to date have been largemouth bass.
Oh, really?
I've heard though from many anglers that we know spotted bass are in the lake and they're caught. We also know from the ecology of those species that the spotted bass tend to stay a little more offshore and the largemouth bass inshore. And so it's not unusual that our boat electrofishing is skewed towards largemouth.
Yeah.
So the bottom line is we haven't seen those species yet. As we start sampling more with a little bit more of our gear types and we get into our spring sampling where those spotted bass will come near shore to spawn, I'm hoping we'll pick up more of those and get a better idea of what the representative mix is. But we just haven't seen many, I don't think any pure spots yet. Yeah.
That's interesting. Just by the way, I know that people have caught white crappie and yellow perch in Lake Toccoa pretty frequently, so I'm sure you're probably at some point gonna, yeah.
It's good to know. Yep. May just be a detection bias at this point.
Hi, Mike Mack, for those of you in the back that don't know me. So I think a lot of us are very concerned about forage out there. And obviously you're gonna be doing zooplankton and then doing chlorophyll A. What about benthic invertebrates, macroinvertebrates? Do you think you will get that with stomach analyses or some other way? Be really curious because you don't seem to see an awful lot of emergence on these lakes, but maybe at times there is some, I don't know.
Yeah, that's a great question, Mike. And so Mike's question was about the importance of macroinvertebrates and insects to the fish. We're hoping that our diet surveys will pick those up if they're a key food source. So we don't have any planned sampling in aquatic vegetation to quantify those densities.
Yeah.
Just due to time limitations and everything else we're doing. The hope was if that's an important food pathway, which after being on these lakes,
Yeah.
I think it may be, you have a lot of natural vegetation
Yeah.
and some non-native vegetation.
Yeah.
So we hope the diet studies will help us get a better idea of how important those macroinvertebrates are.
Do you know if in any studies they have recommended planting of certain types of benthic macroinvertebrates to get them more populous in the lakes?
That's a great question. You know, traditionally, like for pond management in the southeast, to boost fishery production, the key thing to do is actually limit that. If you look in the literature, microfilm won't cooperate.
That's all right.
Really, the recommendation is to promote phytoplankton production.
Okay.
And really enhance the phytoplankton energetic transfer, so phytoplankton to zooplankton, zooplankton to small fish, and then small fish to big fish.
Yeah.
And really, most of the management recommendations focus on having adequate algal shading so that you can minimize aquatic plant growth and make sure those plants aren't competing with the phytoplankton for nutrients and sunlight. But these lakes, I think, they're very clear and pristine water, which is great. I know something the community wants to maintain. So that's definitely an area Dylan and I are going to be thinking about how important these two pathways, the phytoplankton pathway versus the macrophyte plant pathway, and trying to get at with the diets, which one of these might be the dominant energetic pathway fueling the fisheries. So it's a question we hope to get at as best we can, and I think you're right, that's really important, so we're going to try.
Thanks.
Yep.
Hi, Rick Clark. As a lakefront owner on Little Attagahi, first I want to thank you because I have a very vested interest in this whole program. I'm thrilled with what you're doing about the fish. My biggest concern is the sediment. We had a speaker here that started off. It's under my impression that all lakes in North and South Carolina are man-made. Is that correct?
Essentially, they are, the vast
Okay.
majority. We have a few.
And every research I've done for North and South Carolina said that there is a maintenance of sediment removal that is implemented on all these lakes that are in the two states. Is that something that we can get started with here? Because Little Attagahi is in desperate need of a dredging. And we're gonna lose that lake eventually if something isn't done about it. And I didn't know if there was a program or some sort of timeline that could be implemented, say every 10 years or every 12 years, we go in there and we dredge out some of these lakes. The deeper lakes are not issues, but the smaller lakes, Little is in really bad shape, and Attagahi and some of the coves are almost not navigable at this point. So that's my question.
Thanks, that's a great question, yes. So we won't, the science of sedimentation, we could really do a whole project on just sedimentation and get into really a lot of complexity with hydrodynamic models. And we won't obviously go to the level of this project, but we will be identifying areas that are excessive sedimentation. Dylan and I have both, our larger boats won't fit under the bridge to get into Little Attagahi, but we have taken a small John boat just to survey the area and agree with you. You can tell there's been some excessive sedimentation in Little Attagahi. So I can envision us certainly providing some context to experts on something like sediment removal, some private companies who could assist you.
I would appreciate
With
that,
that.
and I'll pass that on to the board.
Okay, sure thing.
Hi, I'm Kim Whalen, and we've owned Lake Attagahi for over 15 years. And I'm just wondering, I've seen more geese there lately than ever. So obviously the concern is the fecal matter and how that's impacting the water and the fish. But also wondering in your studies, how are you able to control for all of the parrot feather mitigation and everything that we're doing, and how are you going to know whether it's naturally something that's in our lakes or whether, those things are harming the fish?
That's a great question. So on the geese side, certainly a concern with nutrients coming in. We will be running a limited number of sediment and nutrient samples to get some baseline. I also understand the community does some frequent monitoring, which you all probably know more than me, for bacterial levels in the lakes. So I think there's already some baseline data that's being collected there. So we'll certainly be making some management recommendations if nutrient levels look like they're exceeding, what's certainly the baseline minimum that's safe in terms of what the states of North and South Carolina recommend for nutrient levels. If you want to drive off geese, there are some techniques out there you can do to haze wildlife or do other things. That's, some places that's implemented because geese can be a concern when there are too many of them and lead to, high nutrient increases and harmful human health outcomes. So that's definitely something we can, recommendations we can include if that's of interest. The second question was about parrot feather, I think. And we're aware of the ongoing efforts to control parrot feather in Atagahi. It sounds like that was a known problem before this project started. And so we won't necessarily be able to control for it, but in our aquatic plant surveys, we at least will be quantifying that and how that differs from the other lakes. And so that's not unusual for a lot of our field studies where a lot of things happen we can't control that are outside of our control. All we can do is quantify it and try to understand it as best we can how it relates to the fishery.
I know somebody had posted, and I don't know, it was just on Nextdoor, so I don't know how true any of it is, but, some people had posted that their dogs had gotten sick from some of the waterways here, so just wanted to put it out there so that
It's a potential issue, sure, yep. Well, thank you.
I'm Rick, I've been here for 21 years now, and fished the lakes, all the lakes. I've read where fish prefer or are more comfortable in a pH environment in the 7 to 9 range. I've never been able to get above 7 in my pH check-in, and it's kind of got the reputation, if you talk to other people, that we got acid water, below 7, 6.5. What are you getting in your initial water sampling pH?
So we are getting levels below 7, and then you're right, that's typical for these high elevation lakes. It's typical, yep. And so just given the underlying geology of the systems, and slightly below 7 is okay. We wouldn't want to get below 6.
Well,
That's right. And so fish, typically the species that we have here, as long as it's above 6, they're typically fine with that. That's enough, for them. You wouldn't want to, go much below 6, but I think we're still in a level naturally that's sustainable. So you're exactly right. pH is slightly lower, but I think we're still in the safe range for most of the species.
Good to hear, thank you.
Dylan, this is for you.
Okay.
Those little chips that you put in those fish, identification,
Yep.
what happens when I catch one and want to eat it?
So, great question. I don't know if I can pull up a picture here still.
But you're gonna,
Yeah, so
are you able to track me
I'll
down?
find you down, yeah. They're entered into the body cavity of the fish. So it's not in the flesh at all of the fish. It's not something that would be eaten anyway. So as you're gutting the fish, you'd be removing that section of it anyway. So.
And if we did come across one, should we give it back to you? Or don't you care?
I'll just say if you found one, it would be useful.
Yeah.
They're really small. They're a little larger than the size of a grain of rice.
Oh.
So
Yeah.
they're small.
Forget it, I'm
Yeah.
done. I'm done.
But Dylan's right. They do go in the body cavity of the fish. They're not in the fillet. So as you clean the fish, if you're removing, all of the guts and everything that you remove when you clean, that's going to you won't have any issue with that. Yep. But great question.
I mean, it's Ray Jackson again. So, but should we save those fish for you guys' study if somebody catches them, or you have enough excess you think it's not going to matter?
I think we probably have enough.
Yeah.
That we'd be okay, but we appreciate the offer.
Yeah. You said you put like a mark on the outside, or there's a mark, or you're not going to tell.
Yeah, there's a bit of a mark, but also as the fish gets older and the day we tagged it is further back, it's tough to tell, yeah.
Okay. One other question. The thermal mixing that you talked about. So I guess at some point that gets to the point where the cold water up top does start to mix, because it becomes more dense. It does start to mix with the bottom surface water. What's the temperature difference have to be, or is that specific, or is it different for each lake depending upon the other nutrients that are in the lake, or how exactly does that work?
That's a great question. So the temperature does have to get fairly close for mixing to occur. Dylan, I'll put you on the spot because I know you were out there measuring temperature in early November. Is that right? As you wrapped up surveys this fall?
Mm-hmm.
What do you see in terms of thermal stratification at that point?
It was close to about mid-50s, I would say, across the whole water column, and that's right when that lake starts to turn over.
I swim in the lakes a lot,
Yeah.
so that's exactly what I noticed. And it was pretty quick, because it was, like 68, 69, and all of a sudden it just switched really quick. Then that must be when that mixing kicks in.
Yep, it's usually right around, for this part of the world, late October, early November. As those surface waters cool, then all of a sudden you get a turnover event. And it can make the lake a little cloudy sometimes, as some of those bottom waters, which have higher dissolved solids, mix with the surface. So if you see it's a little cloudy about that time too, that's another good indication that you've had a turnover, but it's really sudden when it happens. You definitely noticed, I'm sure, yeah.
Do you guys take into account the size of the fish, the condition of the fish around the fish feeders?
Yeah.
I mean, anybody who's just fished around those feeders, particularly the bluegill, they're enormous.
Yeah. It has been something like anecdotally, I've thought of that, and I've got to think about when I'm on the shock boat. And we're doing our standardized transects. I try to avoid that section just so there's not a bias there. But there has been a few times I've gone around. They seem to be a bit bigger, but again, that's totally anecdotal.
But we do have start and stop for each transect, so we'll be able to tell the fish in this transect came from this part of the lake, so it's something we can go back and look at. And if there's a big outlier effect there, we need to be aware of that. Yeah, might not be representative of the population as a whole. Yeah.
Any other questions?
We've got one question. Are you aware of any other prior studies, not as extensive as yours, but data that you might have from the past, that you can determine whether things have improved or deteriorated?
So we are. The question was, are we aware of any prior studies in the past on these lakes so that we can tell if things have improved or gone the other way and deteriorated? So we are aware of several previous consultant studies that have been done on the lakes by private fishery consultants. The Natural Resource Subcommittee was kind enough to send those our way. And so we've reviewed those. I think, all of those studies indicated there may be some issues with potential bass crowding, which is something that Dylan presented and that he's seeing. So that looks to be consistent across the studies. A couple of those studies also conducted water quality transects. So we have some data. Those are kind of snapshots that were once or twice a year, so they're not quite as intensive. Usually taken at just the deepest part of the lake, whereas Dylan's going to do a more comprehensive look across the lakes. But those studies certainly provide a nice baseline that we'll be able to compare to our study to try to understand how the lakes are changing over time. Granted, it's not, like a long continuous study, but we do have these discrete studies that were conducted back in time that we can use as reference. So those are out there, and I appreciate the community sharing those with us.
Dylan, how big's your big bass? Just curious.
That one was 5.5 pounds.
Was that the biggest you got in the lakes?
Yeah, so far.
Yeah, way back in the spring.
Yeah.
I was gonna catch that bass, but you got it before I did.
He's still out there.
I'm just curious about the stocking program, just trying to think about how that factors in. We've been stocking the lakes with trout, right? Or do we stock them with bass? Just trout. And so I would say if we don't stock them for another 2.5 years, it seems like that would be a big impact on, but you'll be able to track what the population is actually doing.
That's the hope, is that we can tell, what the population, if it's declining, we'll see that in the data.
So my understanding is those stocked trout, for example, cannot reproduce. Right?
That's probably right. They need stream habitat, and all the lakes have very little natural stream habitat. I'd be shocked if any of them can reproduce.
Yeah, okay. They sort of, they're genetically.
Yeah,
They are.
Nurseries do that.
Nurseries do that?
NCWRC won't let you stock anything but trissels.
Okay, yeah, all right. Just curious, it'll be interesting to see how the population sort of does over that time.
Yeah,
What are the additions? But I think you need to know that, right?
that's right. That's one of the really.
If we stocked it, then you'd be screwing up your data. Probably worse.
Right. I think not stocking is probably the right thing while the lake study is going on. We had this discussion early on of which, what's the right way to go. But we are really curious to see how those trout that were stocked previously survive. I think that's the big question is, do you have stockpiling of trout, or is their population size getting really large each year? Or are you having a lot of mortality over summer when the water temperature is warm and it really needs that stocking to provide, a fishery in the fall like it does?
I think we're just about at time too, so it's probably a good time if we want to just thank Dr.
Yeah, thanks for all the questions.
You provide mathematics like math, like, you know,